Good afternoon to everybody. We have the pleasure to introduce you, Professor today, Alex Tonks from the University of Cardiff. Alex Tonks leads a research group, a multidisciplinary research group, focused on the identification of molecular alterations, and identification of new targets for myeloid leukemia, particularly acute myeloid leukemia, and through an integrated omics approach that has led him to be focused on several molecules, several proteins that could be involved in also in chemoresistance and clinical relapse in myeloid leukemia. So today he will give us a seminar, and he talks us about his most recent findings regarding redox signaling, proliferation, and metabolism in acute myeloid leukemia cells. Please, Alex, thank you.
Thank you, Michaela. Thank you, Michaela, and pleasure to meet everyone in person here, and welcome to those online. I really appreciate the invite to come here, meet you in person, and your hospitality, so thank you very much. I've got a bit of a raspy voice today, I'm afraid. I think it's the air conditioning of the plane, but hopefully you can understand what I'm saying. So Michaela's already outlined effectively what my mission statement for my group is, which is to identify new therapeutic targets for acute myeloid leukemia. Now, as one can imagine, I approach that through targeting lots of different pathways or cellular functions. But today I want to focus on one particular aspect, and that's redox signaling or oxidative stress. So being a hematologist as well, I'm interested in blood cancers, so that's hematological malignancies, and in particular acute myeloid leukemia. Now, I think I'm right in saying that the audience is a little bit mixed in terms of background and field of speciality, so I thought I'd begin with just a bit of a basic background to hematopoiesis. So hopefully we would all agree that blood is one of the most highly regenerative tissues that exist in the body, and it can generate these blood cells at approximately a trillion cells per day, and these are all derived from this hematopoietic stem cell, or HSC. So what's good about this is that the blood system, for those of you that are interested in solid cancers, represent one of the best models for hierarchical division. So we could expand the HSCs into something in a little bit more detail seen here. And what we're used to understanding now is that these HSCs have self-renewal capacity. So not only do they derive to give themselves, that supports the blood cell production through adult hematopoietic life, but also will also give rise to progenitor cells that you see here. So most of my work will make use of this antigen, CD34. It's uniquely expressed on the hematopoietic stem cells, and by virtue of that we can isolate those cells, and if we can find the right types of tissue like neonatal cord blood, we can isolate those blood stem cells and model normal hematopoiesis, and we sort of call this a sort of ex vivo or a primary hematopoietic model. But in the context of myeloid leukemia, what happens is that these stem cells might pick up a mutation, and that mutation leads to a leukemic stem cell. And unfortunately, the issue with treatment is that chemotherapy is really productive at getting rid of cells, but it also gets rid of normal cells, and unfortunately leaves behind a leukemic stem cell clone. So I think your group, Michaela, and my group have similar goals and objectives, is to try to target this by sparing the normal cells and eradicating leukemia stem cells. So just a little bit about leukemia for those that are not in the know. So it's one of the most commonest adult leukemias, and it has a peak incidence around the age of 85 in the UK. And I wouldn't be a hematologist if I didn't show a Kaplan-Meier curve that we see at the bottom right here. And this is some of the data that has come from the UK NCRI trials. And what we see here is the overall survival of different age groups. So at the very bottom of this curve are the older people, and they generally do much worse than the younger people. And what you see is that a lot of progress has been made since the 1970s to now, but unfortunately even half of the young adults fail to survive over 5 years, and that situation is much more dire in the elderly population. So it's really important that we can find new targets and new therapies. Now, part of the issue for AML is for treatment, that is driven by the highly heterogeneous molecular nature. And what I mean by that is this slide here. And I don't want you to read too much into it. It's just meant to demonstrate that acute myeloid leukemia has a number of molecular aberrations, indeed over 100 of them. And some are more frequent than others, and that's shown here in this little graph at the top. But we can also classify these leukemias not only based on their cytogenetic abnormalities, but also on their sort of favorable risk outcome, if you'd like. So some mutations like 821, which is called RUNX1ETO, has a more favorable outcome than those that you see at the bottom here. That would be patients with complex karyotypes or cytogenetics. So this really hampers how we can treat AML. So we need to identify new novel targets, and to some extent this paradigm has already been proven. And these agents on the left, they're a bit old now, I would say, because I think between 2018 up to now, there's been a big shift in targeted therapies for AML, such as gilteritinib, Mylotarg, lipid-encased drug delivery systems. So I think there's hope on the horizon that the targeted therapy approach is a valuable approach to take. But we do need more targets, and I don't think they're going to come from high-frequency mutations, because I think they've already been detected somehow. So maybe we're looking at alternative splicing, protein isoforms, or epigenetics. And I think it's fair to say that AML is a disease of epigenetics. So one way to identify new therapeutic targets is to model them. And I want to go back to that CD34 system that I use, and this is maybe for some of the more early career researchers in the department, because I'm sure many of you take a similar approach. So how can we model them? So we take one of those abnormalities. For 20 years or so I've worked on RUNX1ETO, but I've also been working on mutant NRAS, and NRAS today will be a common theme through the presentation. And then we try to overexpress that oncogene, if you like, or a target of interest into CD34 positive cells. And we do that using retrovirus or lentivirus transduction. And these vectors, they encode a selectable marker, for example, green fluorescent protein or DS-RED. We can also do it in cell lines, but cell lines are not that tractable, I would say, for differentiation analysis. So the primary hematopoietic model is particularly useful for differentiation and self-renewal, but of course we can read out all these other endpoints that are of use for us. Having established a phenotype, maybe it's anti-proliferative or increased survival, whatever that would be, we then go on to look for mechanisms. And we generally have done that in the past in two different approaches. Molecular endpoints, such as using, well, back 20 years ago, microarrays, more recently, next generation sequencing. And the other approach is more proteomic, because in my experience, 60% of the changes in transcription don't equate to changes into protein expression. So we like to try to take both approaches and platforms to get as much information as possible. It's not to say one is more valuable than the other, but you get a lot more added value by taking in both these approaches. We've tried to use SWAF mass spec, which is a mass spectrometry technique that can look at absolute quantitation of proteins, or we use iTrack, where we can look at relative abundance of different cells, of different proteins within different contexts. So hopefully we then identify a mediator of that effect. And in the example I'm going to show shortly, reactive oxygen species was one of those. So we can then go back to the model system and see if we can recapitulate the effects we've seen of the original abnormality. Maybe we can knock down, overexpress, and hopefully we can provide further evidence that's important in mediating the leukemia phenotype. So one of the strengths at Cardiff was sort of this orange or yellow coloured box, I would say. Cardiff was one of the UK centres for receiving AML blast samples from the UK NCRI clinical trials. And those samples banked came along with clinical information. And that matched data was highly important in terms of trying to validate the role in disease and provide evidence as a value as a prognostic marker, for example. To some extent, we've lost a bit of that unique selling point, because you can now go online. CBioPortal, I think, is a particularly good platform if you haven't used it, but you can download expression data, correlate it with clinical information, but what it doesn't give is the matched AML sample where you can do further studies. And that's what we tend to do. We then take those potential targets and we take our AML sample and we see if we can change the proliferation or survival of AML blasts. And that leads us towards more preclinical evidence for a clinical trial. So we're currently at the stage of looking at a number of different targets at different stages in that pathway. CD200 is sort of going forward towards trying to find funding for a clinical trial. But today I'm going to focus on this part, which is reactive oxygen species driven by this enzyme here called NADPH oxidase, or NOx. So we've used different platforms. This is sort of what my group does, and if anyone's interested in talking about any of these other targets, I'm more than happy to discuss. So targeting ROS in acute myeloid leukemia, I've been doing this for several years now with a co-PI called Richard Darley, and Paul, Andrew and Abdul are PhD students in my lab that have contributed enormous data, I would say, over the last 10 years or so that have contributed to many of the streams of research that we're undertaking. So background to ROS and how my ROS is different to your ROS, then we'll talk about it. But first, some basics. Molecular oxygen basically essentially gets metabolized to superoxide, and there are two types of sources in general that would lead to this. One is the electron transport chain, complexes 1, 3, maybe even complex 2, and the second one is this NADPH oxidase, or NOx, that oxidizes oxygen to superoxide. You then get subsequent reactions. Superoxide dismutase, seen here, catalyzes the reaction towards hydrogen peroxide, whilst a phantom reaction can also occur with iron or some other molecules to give you hydroxyl radicals. And there are lots of splinter chain metabolism that goes on there that gives other free radicals. For example, I'm not going to talk about it, but hypochlorous acid or bleach. But for today, I'm going to focus on these two here. That's what I mean by the term ROS. So we can use different tools to measure or understand more about ROS signaling, and these are some of the agents that we've used in the lab: apocinin, DPI, VAS, or more recently GSK, which is a more specific inhibitor to NOx. So of course, to understand the role of ROS in health and disease, we need to have some essential knowledge of the spatial and temporal production or characteristics of ROS. So NOX2 is membrane bound. It's in the plasma membrane of many different types of cells, and I'll come on to that shortly. And as I said, produces superoxide that converts to hydrogen peroxide. And there are different types of molecular probes or chemiluminescent probes that can measure or quantify superoxide. And the one we tend to use is Diogenes. This is based on luminol, but you can also use other probes like Amplex Red that will bind specifically to hydrogen peroxide. And really, I think it's the availability of different probes that hampers reactive oxygen species research, because there are a number of positives and a number of negatives to the different types of probes that are out there. But what can happen is that hydrogen peroxide can traverse the plasma membrane. We think it could diffuse. I'm not quite sure that is the case, but there are evidence of aquaporins which transport hydrogen peroxide to the intracellular compartment of the cell. I guess it's up for debate how far hydrogen peroxide through this process goes into the cell. It's quite a journey before it gets detoxified. But there are other routes of hydrogen peroxide generation, as I said, the electron transport chain, or indeed NOx isoform members are distributed in different intracellular subcellular compartments. So NOx4, for example, is present in the nuclear membrane or endoplasmic reticulum. So there's about 7 of these family members, by the way. 5 of them are NOx related and 2 of them are DUOX1 and DUOX2. But let's just focus on these NOx isoforms. So what you'll see here about NOx2, the reason it becomes active is because it has these intracellular signaling molecules. Once they get phosphorylated, they change their conformational shape and move to the plasma membrane to constitutively activate NOx2. Conversely though, NOx4 doesn't require those subcellular compartments and can produce hydrogen peroxide without any additional cofactors. But this is the interesting thing. NOx2 was really developed as a phagocytic defense in response to bacteria through that phagocytosis when phagocytes, neutrophils or macrophages, for example, would engulf bacteria. But a seminal study, I think, by Piccoli showed that these NOx isoforms are present on hematopoietic stem cells. And you'd have to ask yourself, why would that be? Because these HSCs are not circulating surveilling for infection, but in fact reside in the bone marrow. So it doesn't seem to play a logical role in innate immunity. And so it's argued that perhaps they're playing a more signaling role. And I think that's well accepted now, that NOx or reactive oxygen species can play a number of roles outside of innate immunity. And today I think I'm going to focus on regulation of proliferation and obviously in the context of cancer or AML. So this is a bit of an oldie but goodie review. I still quite like it. It shows how ROS has been implicated in many different aspects of cancer development and how the fine balance is absolutely required to either increase cell survival, promote cell death, for example. And one of the early studies that showed this was by Irani in 1997, where they took fibroblasts and transduced them with RAS. And what they were able to see was this incorporation of thymidine, suggesting an increase in proliferation due to activated RAS. Of course, that doesn't tie in reactive oxygen species, but when they cultured those cells with N-acetylcysteine, they were able to see a reduction in that mitogenic effect or proliferative signal. So that was some of the first evidence that suggested reactive oxygen species can change proliferative responses in cancer cells. And then in 2007, Padua et al. Looked at an in vivo model of this, where they basically created an in vivo activated NRAS model and looked for DNA damage. And that's what's shown on the top. So this is sort of the untreated controls, and when you treat them with N-acetylcysteine, the number of DNA damage molecules that are occurring is less. Again, showing a protective effect of NRAS and antioxidants. So that evidence led us to ask 3 questions. Do AML cells alter their production of reactive oxygen species, and in particular we're interested in NOx2? Does the production of ROS impact hematological cells, whether that be development, proliferation, differentiation? And more importantly, can we manipulate ROS as a means to eradicate leukemia stem cells or modulate hematopoietic development? And for the most part, I think we've been able to answer 1 and 2, still struggling with number 3, but that's sort of work ongoing within the lab. Okay, so starting with, do AML cells alter the production of reactive oxygen species? So we take our CD34 cells that we've isolated from neonatal cord blood and we transduce them with our vector expressing NRAS or HRAS, and we're able to observe a transduction frequency in our system of pretty much over 70%. This negates the need quite often to do flow sorting, and we can go straight to flow cytometry or other cell biological endpoints. But just because they express GFP or DS-RED, it doesn't mean they express the transgene of interest. So it's always important to validate whether that's the case. And whilst we can't show activated MRAS, we can show that we do get more MRAS protein. So question 1 was, do NRAS result in HSCs producing more reactive oxygen species? And I think you can see that here. So there's a dose-dependent increase, probably on average in our studies around 6 to 7% when we use HRAS. NRAS is a little bit more clinically relevant in terms of the mutations that we see within the clinic. So we know that ROS levels are increased, but how do we tie that back to NOx? So this is our control here, and when we incubate with DPI, the NOx signal is completely inhibited and the production of ROS is ablated. And this is also true for other agents that inhibit NOx2, like apicinin. But importantly and differently to some other studies, we use rotenone, which is a mitochondrial poison, and we don't see the ablation of that extracellular signal, suggesting to us at least that the ROS we're detecting is extracellular, and that ties in with the luminol probe, because luminol is restricted from entry into the cell based on the chemistry. But we still wanted more evidence for NOx2. So we looked for the expression of NOx2 on the cell surface of HSCs infected with RAS, and you can see here that we've seen maybe a 2 to 3-fold increase in expression of NOx2. We also went to an in vivo model where we isolated stem cells. In this case, they're called SCAR1 positive cells, and we isolate them from a NOx2 knockout mouse. And when we did that, we failed to detect expression of extracellular ROS using luminol-based assays. And then finally, we went back to those cofactors to see if there were changes in expression of P67, P47, or P40-FOX. And if you remember, these are the ones that are required to activate NOx2. And what you can clearly see in the RAS side of things is increases in P67 and P40-FOX, and to some extent RAC, which is a stabilizing protein. So all our data so far suggested that NOx2 was the primary source of reactive oxygen species, at least in our CD34 positive cell model expressing mutant RAS. But how does it impact upon the cellular function? So one of the things we looked at was cell cycle and proliferation. So we did use PI to measure DNA content, and you can clearly see that the RAS transduced cells are in a much higher level of cell cycle. And when we incubate with low concentrations of DPI or N-acetylcysteine, we can revert the effect that we see in terms of proliferation. But all that was about our human primary cell model, not necessarily leukemia. So more recently, in some unpublished data here, we've looked at cell lines, AML cell lines, about 13 or 14 of them, and we've assessed the production of reactive oxygen species using that luminol probe. And what you can see here is a variable level of NOx-derived reactive oxygen species. For example, THP1s produce a moderate level of ROS, whereas MV411s we sort of term underneath background or low levels of ROS. NOMOs are quite interesting. They produce magnitudes of order of ROS, and we're not really quite sure what that is, probably related to the genetic background of that cell line. But we can use these two cell lines as an example to do further studies, one as a ROS producer and one as ROS null. So if we take the ROS null system, we want to give it ROS. There are a couple of ways you can do that. You can give it a bolus amount of hydrogen peroxide, but that's quite a large amount to give and not sustained over a period of time, so we don't tend to do that. But we make use of this enzyme called glucose oxidase, and this converts glucose or oxidizes glucose, and the byproduct of that is hydrogen peroxide. So we measured hydrogen peroxide using Amplex Red, and correlated the amount of enzyme units to see if we had a linear response, which of course we have. And we generally incubate between 5 and 20 milliunits per mil of GOX in order to have a continuous system of hydrogen peroxide within cell culture. And when we do this, this is what we observe. We observe an increase in proliferation of our MV411 cells. If you remember, they're the cells that don't produce ROS, but if we give them ROS, they proliferate. And importantly, if we include catalase in that culture condition, which is an enzyme that catalyzes the breakdown of hydrogen peroxide, we can blunt the response further. So we can also do the opposite. We can take our ROS producing cell line, THP1, and either use CRISPR or shRNA and knock down NOX2 in those cells. So the blue histogram that you see here is the expression of NOX2, and the light pale blue is the sort of removal of over 90% of that protein in those cells. And when you do that, you can also look at the functional aspect of that, and you can see that the production of ROS in those cells is now lower than our cells treated with the NOX inhibitor DPI. So what happens to proliferation then? Well, again, what we observe is a reduction in proliferation due to the loss of the ability to produce reactive oxygen species. But whilst we move forward to AML cell lines, it doesn't really tell us about primary AML. So Paul spent a fair few months, several years ago now, looking at the production of reactive oxygen species from primary AML blasts using that Diogenes assay. And what you can see here is, in essence, is over 60% of the AML blasts produce higher levels of ROS compared to our normal HSCs. And I also want to draw your attention to the scale, because this is a log scale on the side. And what that means is that actually you've got a fair proportion of these cells producing really high levels of reactive oxygen species. And to some extent, that does mirror what we see in the AML cell lines. We haven't been able to attribute whether this variability in ROS production is associated with particular cytogenetic abnormalities in AML. There's just not enough power in this analysis to do that. But there was a recent paper that looked at what I want to say is an outdated classification now, the French, American, British classification. We still use it in terms of looking at hematopoietic cell differentiation. So in the FAB-M4, M5, that's a more differentiated AML versus the M0. And what they showed was higher levels of ROS tend to be observed in the more monocytic macrophage type AML, which would go towards its sort of role in innate immunity. And incidentally, that's something we also observed several years earlier. Okay, but what about proliferation? So this chart's perhaps not well displayed here, but what you can see in these lines here is that AML with low ROS and given GOX proliferate much more than our control HSCs that are observed down here. So I think, to summarize, that's that part of our work that took about 6 years and resulted in 2 papers. I've convinced you that RAS promotes reactive oxygen species via NOX2, and that's where most of our research is now focused. I didn't show you this, but we were slightly disappointed. We were rather hoping that ROS would change the developmental program of hematopoietic cells, in that they would differentiate differently, but we didn't observe that. Really, the only phenotype we did observe is changes in proliferation or survival. And to support the proliferation studies I didn't show you today, but we do see increases in cyclin proteins or cell cycle inhibitors. Okay, so what about understanding how reactive oxygen species have done that? And this is what my lab has focused on in the last 5 years or so. So we were talking about this earlier, but redox sensitive cysteine residues exist in many different types of proteins. And it's the exploitation of this characteristic that allows us to think reactive oxygen species or hydrogen peroxide can act as a signaling molecule. So for example, if hydrogen peroxide's inside the cell, it can affect these protein tyrosine phosphatases. And these are cysteine sensitive redox residues. So hydrogen peroxide would oxidize this, inactivate the PTP, which would then switch off the signaling activity. And what makes it a signaling molecule is that these sensitive residues can then sort of be regained or reformed, if you like, back into an active component. So hydrogen peroxide can signal through this type of mechanism. Alternatively, there's some evidence that reactive oxygen species can stabilize transcription factors like HIF-alpha, for example. And there's a lot of interesting evidence about the role of HIF in normal hematopoietic stem cell development. So this is just an example down here showing the concept, if you like. Transcription factor has a redox sensitive residue. ROS can interfere with this, which then stabilizes or destabilizes the transcription factor. That leads to a change in gene transcription. So that was the idea behind our original sort of mechanistic studies. So Andy took human CD34 cells and the MRAS model and transduced these cells and asked the question, are there transcriptional changes in reactive oxygen species? So the first thing to do, which was done by Lucy, was to again confirm in those models, in that model, and before we send the cells off, that there's an ablation of the ROS response. And this is important because I want to drive your attention towards this type of analysis. So the first bit is looking for gene transcription differences between control and NRAS. And these changes will be a mixture of what we call RAS and ROS dependent changes. And then in the presence of DPI, we need to see what genes are sort of being changed back, if you'd like. And by observing those, we can then say these are the ROS dependent gene changes. And when we took that approach, the long and short of it is we identified 23 ROS specific target genes. I think I was hoping for a bit more, but I was happy with 23, and when I did the pathway analysis, I was even more happy that actually a lot of the changes were related to metabolism. And indeed, it fell down to two different places, at least in the gene transcription area. One of them was glycolysis or changes in carbohydrate metabolism. So that's what's led us down this story for metabolic studies. So if you don't remember your basic biochemistry, glycolysis is the 10-step conversion of glucose to pyruvate, which then goes into the Krebs cycle. And these little orange arrows indicate the increases in expression of genes that we think were ROS sensitive. And the sort of raw data, if you like, is shown for that on the side. And let's just pick GLUT3 as an example. GLUT3 was significantly increased in response to RAS, but was reduced when you included the inhibitor DPI. So reactive oxygen species in our hematopoietic cell model is increasing a number of gene expression products related to the glycolytic pathway. But going back to that foundation model system that I showed you, the flow chart, we need to bring it back to AML patient samples and make sure we can tally up the primary model, the AML cell model, and AML patient blasts. So we went to cBioportal, downloaded the gene expression data for any gene we think could be included in carbohydrate metabolism, and then stratified the patients according to whether they have high NOX2 or low NOX2. Now ideally, in an ideal world, we'd want to measure reactive oxygen species in these, but we can't. But our previous studies suggest that the expression level of NOX2 is or correlates with the production of reactive oxygen species. So, take this with a little pinch of salt, I think, but I hopefully you would agree that the majority of high NOX2 patients have a signature that has increases in gene expression of genes related to carbohydrate metabolism, and that's these sort of 8 or 10 here. So in all the cases, the red dots are higher expression in AML or AML with high ROS versus AML with low ROS. So as I said a while back now, I think gene changes don't always equate to changes in protein and doesn't necessarily equate to changes in activity either. So we looked for glucose consumption being the principal step, if you'd like, in glycolysis. So one of the ways to do that is to use a molecular probe called 2-MBDG. And what happens is it gets taken up into the cell and converted to a fluorescent readout. And we can then measure that readout. So underneath, you see the sort of raw data for this. So our black chart, our black bar, is our NRAS cells. our control cells, when you look for increases in glucose uptake, you can see it increase by 20 to 40%. But when you include VAS, which is an inhibitor of NOX2, you get a reduction in that glucose uptake. So our hypothesis sort of suggested that NOX2 leads to increases in extracellular ROS that leads to increases in glucose uptake that support proliferation. And we were hypothesizing that changes in glucose metabolism might support these proliferative responses. So to do that, we took 20 AML patients and measured reactive oxygen species on them. This is the traces for luminescence. And quantitated that into a single bar point. So we can either look at rate or peak light production. And then took the median of those values. And anything above the line was the high ROS group, and anything below the line was the low ROS group. Remember, these are AML patient blasts. So we had 10 in the high, 10 in the low, and we used 6 normal controls or mononuclear cells. Now, we didn't have academic partners to look at the whole metabolome of these cells, so we outsourced them to a company called Metabolic. It's a US company, and they used a combination of liquid chromatography and mass spec and identified 444 named biochemicals. And I was quite pleased when I seen this PCA. When we put the PCA plot together, what we could see is the controls seen in these white circles distinctly separated from the AML samples based on their changes in biochemicals. But what was interesting was that for the most part now, the AML high red triangles separated from the AML low ROS, not AML low, ROS, AML, ROS low. I'll get there, Michaela, right? The two that were borderline were the two right on the line of this median, of this sort of median analysis. So, you know, we didn't, what's the word? We didn't make this up. This is how it came out. And I was, you know, quite interested that these two are potentially ROS low, really. But we focused onto glucose metabolism, and we looked for the changes in the biochemicals in this pathway. And for the most part, everything that was proximal at the top here changed. We've seen higher levels in the ROS AML high than we did in the ROS low group. And in fact, after that, we didn't really see any significant changes below that. So we were quite interested in that to begin with. But what's important about glycolysis and this particular point here, it's a branch point for other metabolic pathways. And one of them includes the pentose phosphate pathway, hexose monophosphate shunt, and serine synthesis as well. So to me it seemed like reactive oxygen species were leading to an accumulation of these early to intermediate carbohydrate molecules, if you like. What's also interesting, and we can look at, because we did whole metabolome, we could look at all the biochemicals that are associated with these pathways. And indeed, you get to see higher levels of biochemicals involved in nucleoside synthesis. And this would make sense. If you need to proliferate, you need more nucleotides. And also what we've seen was increases in glutathione or GSH. Again, interesting from 2 points of view. One, GSH supporting proliferation, and two, GSH is an antioxidant. So, you know, I'm not really quite sure what it's doing. Is it producing more GSH to support proliferation, or is it producing more GSH to help detoxify the AML because it's undergoing oxidative stress? So we wanted to try to understand this mechanism further, and to cut a long story short, I don't want to show you all the Western blot data, but we went back to that hematopoietic model with NRAS and Western blotted every protein in the carbohydrate pathway or the glycolysis pathway. And the only protein that changed when you gave it RAS and was removed when you inhibited was this protein here called PFKFB3. And PFKFB3 sits in this part of the pathway. I'll zoom in for you. So if you didn't know, it's got 4 isoforms. Number 3 is the one that has more kinase activity than anything else. So what happens is fructose 6-phosphate gets phosphorylated by PFKFB3 to fructose 2,6-phosphate, and that then activates PFK. That then leads the carbohydrate, the carbons through the glycolytic pathway. So we needed to determine and provide some more evidence that NOX2 derived ROS can modulate PFKFB3 protein expression in AML. The data so far is in our primary model. So we went back to those cell lines. Remember, THP1 was ROS high, so they naturally have a certain level of PFKFB3. And when we knock down NOX2 or inhibit it, we can lower the levels of PFKFB3. And also, as I've shown you previously, we reduce proliferation, and now I'm showing you we can reduce glucose uptake. And we do the converse, where we take the ROS low and we incubate with GOX, and we get this dose-dependent increase in PFKFB3. And that again is associated with increases in proliferation, which I've shown you previously, but now we're showing we got increases in glucose uptake. So I think it's fair to say that PFKFB3 overexpression and knockdown changes proliferation and glucose uptake in AML cells, but we really don't know how that is done. So the aim was to provide more direct evidence. So we took MV411 cells and overexpressed PFKFB3 itself. And when we did that, we got an increase in relative proliferation and an increase in glucose uptake. And again, we took THP1 cells and we knocked down NOX2, shown here. And what we had is a reduction in proliferation, but no changes in glucose uptake. And unfortunately, I don't think I can explain why that is. I was a bit disappointed we didn't see a reduction in glucose uptake, but maybe the cells have enough glucose to support themselves at those time points that we analyzed. So a postdoc in my lab, Namrata, she went on to determine, and this is more recent data, how PFKFB3 is regulated by reactive oxygen species. So AMPK can lead to changes in PFKFB3, which leads to changes in glycolysis. So one of the first things we wanted to do was to take our MV411 cells, incubate with GOX, confirm PFKFB3, but check for AMPK levels. And when we look at phospho-AMPK, we see increases in phosphorylation of this protein, suggesting increases in activity. And also in the literature, uncoupling protein 2 has previously been shown to stabilize or activate AMPK in this pathway. So we also looked for UCP2 expression and again seen an increase. Now we wanted to inhibit UCP2, but we really couldn't find a decent inhibitor. But there was an agent called genopin, which destabilizes UCP2 to sort of give the same endpoint. And when we did that, we can reverse all the effects that we've seen related to ROS affecting AMPK, PFKFB3, and glycolysis. And then the last bit of data I want to show you related to that was that another postdoc in the lab, Nader Omidvar, he took our THP1 cells and put them in vivo. So this is the model that you have here. And if you're not familiar, what you got is basically a transplantation assay. So this population here is your tumor, and when we give PFKFB3 knockdown cells, we don't establish tumorigenesis in the mouse, suggesting that PFKFB3 was required for that effect to occur. So this has sort of led us to a point now where we think either glycolytic inhibitors or inhibitors for NOX2 might be a potential target for therapy in AML or certain AMLs. So does one really want to inhibit the metabolism? That's the question. And early studies would say, I don't think you should go that route. But more recent studies have shown that actually there's a therapeutic window that we can target. And glycolytic activity has previously been linked to poor prognosis and therapy resistance. So we think there's some evidence that will allow us to go forward with this route. And the last bit of evidence is that a study by Chen showed that AML cells were sensitive to inhibition by a sort of broad spectrum glycolytic inhibitor. So I'm hoping that we can see more specific and more fine-tuned metabolic inhibitors like inhibition of PFKFB3, for example, and they might work better plus or minus our NOX inhibitors, for example. So some of my earlier studies used this agent here, 3PO, as an inhibitor of PFKFB3. I'll show you that data in just a second, but our unpublished stuff is now working on this AstraZeneca inhibitor, so it's a bit more specific for PFKFB3, and unfortunately I haven't got any data to show you today. It hasn't come off yet, but I'm hoping we'll see what I'm going to show you now with 3PO. So 3PO is an inhibitor for PFKFB3, and when we take our MV411 cells and incubate them with GOX, we get this increase in proliferation. And when we use 3PO or PFK158, which is another inhibitor, we reduce that proliferation. And we do similar studies in THP1. So I think we're at a point now where we need to do this in primary AML patient samples, and that's the work that's currently ongoing in our lab. So the future perspectives for us is inhibition using AZ67 in combination with this NOX inhibitor, and these two are sort of pharmacy grade now, or pharmaceutical grade inhibitors. And then the next step after that is to go in vivo. But obviously one of the things we don't know, and this is a big aspect of our current work, is the genetic basis of heterogeneity in AML according to ROS. So we're going to explore the sensitivity of NOX inhibition in primary AML and see if we can observe a genotype that's particularly sensitive to NOX2 inhibition. And then we're going to identify a sort of synthetic lethality primary AML screen that if we knock out a gene and has additional effects along with NOX2, maybe we'll have a more effective therapy for patients with AML. So in conclusion then, so the oxidases have a spatial and temporal sort of production of reactive oxygen species, and we see that in about 60% of AML. They have a diverse range of effects, and we've focused on proliferation today. We've shown that you get increases in glycolytic gene changes and functional activity, particularly with increases in glucose uptake and changes in carbohydrate metabolism, that have led to increases in proliferation and survival. So we're hoping to use inhibitors to NOX2, antioxidants such as catalase, or inhibitors to glycolysis to augment each of these responses. And I didn't have time today, but there's also cell cycle arrest and promoting cell death that would also be important. So this is sort of a graphical abstract of what I talked about today. Hydrogen peroxide leads to changes in UCP2, which increases phospho-AMPK. And I haven't shown you, but there's some data about mTOR and nutrient sensing with reactive oxygen species that would be important. And of course, all the changes in carbohydrate metabolism. Now, one aspect of that metabolic study we did was changes in amino acids and changes in lipid metabolism. And actually, changes in lipid metabolism were the most significant changes in our data set, and highly reproducible, and lots of different polyunsaturated fatty acids. So going along with some of your work, I think about how reactive oxygen species can lead to lipid peroxidation is something we're investigating in our particular studies here. So finally, I think I would just like to acknowledge lots of group members. I tried to highlight them as I go along, but in particular Richard Darley, who's been sort of co-PI on many of these projects with me. And just to show you that Cardiff is not always doom and gloom, it is often nice and bright and sunny. Not today though. I'm glad I'm here. Thank you. Happy to take questions.
Thank you, Alex, for this very, very interesting, insightful seminar. Is there any question from the audience?
Rafaela.
Thank you very much. It's very, very interesting. I have a question. Just what did you serve about transcription factors? Because I understand that it is the uptake of glucose that is the major determinant for the increases glycolysis and perhaps antioxidant like GSG, etc. But I don't feel is transcription factor related. This maybe also microRNAs or something like that.
Yeah, do you know what? I completely agree. Let me see if I've got this here. Oh, that was my, not that one. I'll keep going here. So we thought HIF alpha might be interesting, right, to see that. But exactly what you said, we don't see changes in HIF alpha. So the signature that we observed through the glycolytic mRNA gene transcription changes were very much a HIF alpha change. But when we try to measure it here, we don't see that. So this is the change in mRNA for HIF alpha, but when we look at PFKFB3, there's no change in it when we knock down HIF alpha. And these are just some controls to show that we were measuring HIF alpha. So, you know, I think I agree with you. And we then also looked at other bona fide transcription factor sensitive, ROS sensitive proteins and didn't see any changes there. Things like, I don't know, FOXO3 or something like that. And we see no changes there. What we do see is changes in signaling molecules. Things like P38 MAP kinase, for example, and how they sense reactive oxygen species. And I think partly that might explain why we didn't see so many gene changes either. So whilst we've seen lots of 300 gene changes in the RAS model, only 23 were ROS specific. And out of those, only one of them really changed in terms of protein. So, I think I would agree with you. I don't think the transcription factors directly with ROS are playing a role. Maybe indirectly through signaling or something like that might be possible.
Okay, thank you.
Is there any other question? I have a question regarding regarding glycolysis. You talked about glycolysis, but what about oxidative phosphorylation? Is there any correlation between these two? That means acetyl-CoA is driving to...
So some of the work we wanted to do was to go use the seahorse, right? And maybe do more global analysis that way, and we couldn't get to do that. The alternative approach, we wanted to do carbon tracing, and we really couldn't find somebody that could trace these carbons through the loop for us in this particular model. So we really only ended up doing, analysis at a single time point of those metabolic, of those biochemicals. And then when we looked at ROS high and ROS low, there was really no changes in the mitochondria or in the Krebs cycle of those different biochemicals. I think, though, AML overall had more mitochondrial activity, but didn't really differentiate ROS low and ROS high.
This is related also to the amount of superoxide.
Yeah.
That means that somehow it depends mostly on cytoplasmatic ROS production, superoxide production, more than from mitochondria superoxide production.
So a long time ago, we looked at mitochondrial superoxide and really couldn't see any differences using a probe called Mitosox. So I think most of our work has sort of negated the mitochondrial ROS side of things. Doesn't mean there's no intracellular ROS changes going on. And I think I said at the beginning, I think having the right probes to measure oxidative stress within the cell is important. And there are molecular probes like plasmids that are ROS sensitive that you could use, but they're very sensitive to pH. So we haven't really had much success with those either. So at the moment, we've stayed away from the inside cell and stuck with outside ROS. But yeah, I think you're correct. And of course, if we've got changes in lipids, there are other enzymes like lipoxygenases that can also lead to increase in reactive oxygen species or oxidative stress. And there's also something called xanthine oxidase. So there are other mechanisms of producing ROS. So I think subcellularly, the context will be really important. Where is it produced and when is it produced?
Yes, absolutely. So it seems to me that the glucose uptake, the increased glucose uptake, could also be used, as you said, to fuel the pentose phosphate pathway, particularly for nucleotide, but also for NADPH production. So to counteract the excessive ROS production.
Yes.
It could be a mechanism to balance the excessive ROS
I think so, yeah. And we sort of knew that with the GSH a long time ago, because we measured GSH through, I don't know, biochemical means, if you like, not through mass spec or anything like that. And we already seen increases in antioxidants as well. So catalase, for example, not so much many of the other typical antioxidants like peroxyreductions, but peroxyredoxins. Really, catalase and GSH was what we observed. But you're right, I did chicken and egg. Which one is first?
Which one is first?
Yeah.
Okay. Is there another question? Also from from the remote audience. Not at the, no? No, okay. So, thank you again, Alex, for your excellent presentation. And
Pleasure.
I wish,
Thank you for listening.
thank you for coming. Okay.